Okay, this video is going to go over the macromolecules category of lipids. And again, this is for an AP or an IB biology course. So some general information about lipids. Lipids come in different categories. So lipids is the broad category, but in common speaking, we tend to just say fats or oils. But just note that these, if you say fat, it does not apply to all lipids. The same thing with oils. These are subcategories. So there are fats, there's oils, there's waxes. There's also the triglycerides. Those will be in the fats or oils. Then we also have phospholipids and steroids as well. So in this category, these molecules are made out of only carbon, hydrogen, and oxygen, very similar to how carbohydrates were. These are for long-term energy storage in organisms, as well as for insulation, which would be like heat regulation or chemical insulation, if we're talking about the nervous system. This category does not actually make a true polymer. We do have some smaller molecules that will covalently bond and make some larger molecules, but they don't form this extensive, infinitely long chain of molecules. So let's talk about the triglycerides first. These are made of one glycerol molecule, which you can see here. It's 3 carbons and it has 3 hydroxyl groups on it. And then it's a triglyceride because it has 3 fatty acids. And you do need to know all of this information for your exam. So again, these molecules will undergo dehydration synthesis and you'll get 3 waters being produced. And you'll get a triglyceride, and it kind of looks like a letter E. You have the 3 fatty acid or the horizontal bars of the E, and then the glycerol makes up the vertical bar of the E. And where it says ester linkage, this is a special organic chemistry name for the covalent bond in between these 2 molecules. We had the same in carbohydrates, that their special name is called a glycosidic linkage. Nucleic acids, it's a peptide linkage or a peptide bond. For proteins, peptide linkage or peptide bond, and then for nucleic acids, you have something called a phosphodiester bond. That information is extra. That's not on the exams. But for triglycerides, these are what make up your fats and your oils. Okay, there's 2 different kinds. There's saturated versus unsaturated. So a saturated is a fatty acid that has absolutely no double bonds at all. So in the example picture here, the one on the left, palmitic acid, is a saturated fatty acid because there are no double bonds. It is fully saturated with hydrogen. It has the maximum number of hydrogen atoms it can possibly have. Unsaturated has at least 1 double bond. It can have more. So you can actually have a monounsaturated or a polyunsaturated. So this unsaturated doesn't have the full complement of hydrogens possible, and actually the double bond it's a stronger bond, and it will physically change the shape. Right here you can see it on the right highlighted. It will actually bend the fatty acid. Okay, and actually unsaturated fatty acids are healthier for humans than are the saturated ones. Saturated fatty acids, because they're straight, they can pack more easily and kind of stick together, and that's what makes fats, they're solid at room temperature. So this would be like butter, lard, animal grease, stuff like that. But an oil is liquid at room temperature, like an olive oil or a canola oil, and that's because of that bending from the double bond. It allows the molecules to slip and slide past one another. They can't tightly pack like that, so they stay liquid. And those are actually much better for your cardiovascular system health. Now, phospholipid is a modified triglyceride, so it still has that glycerol backbone, and it will still make those ester bonds by bonding with fatty acids, but in this case, it will only be 2 fatty acids. And in the third place, you'll actually have a phosphate group bond along with a choline head. The phosphate and the choline together make up the hydrophilic head of the phospholipid. Fatty acids can vary in the number of carbons that they have and the number and placement of their double bonds. You are not required to know all those differences, but just know that they are different. Okay. Um, the phospholipid is a very unique molecule. It's very important. We're going to come back to this in a whole other chapter on cell membranes and membrane transport. But the phosphate and the choline head are actually polar. They make the head of the phospholipid hydrophilic, meaning it attracts water. But the fatty acids, most lipids, with the exception here, lipids do not dissolve in water because lipids are hydrophobic. They're not polar at all. So the fatty acid tails are actually nonpolar. So you have this molecule with a polar region and a nonpolar region, and when you have that in the same molecule, we call that molecule amphipathic. It goes both ways. And this phospholipid will arrange itself with many other phospholipids in a bilayer. So the phospholipid will actually, so there's the cell membrane, and all of the organelles within it are made of membranes too. It will form this complex structure, which we'll come back to later. But here's at the base of it is the phospholipid bilayer. So you'll have a phospholipid, it kind of looks like a little bobby pin, and then its mirror image will be below it. So or kind of like, on a sandwich, you have two layers of bread. So you have two full layers of these phospholipids that go from top, to bottom, and that's what makes up every single cell. Every living thing is made of cells. All cells must have cell membranes. Even prokaryotic cells have cell membranes, and they're all made of the phospholipid. And our last category is steroids. Steroid is a structural name. Okay, the structure of it is what it looks like. It has this shape with the 3 fused hexagon rings and 1 fused pentagon ring. What makes the different steroids different is their functional groups. Okay, and oftentimes our bodies, our liver especially, can convert one steroid to another back and forth depending on what's needed at that moment. But what steroids do as their job, their function, they function as a hormone, which is a molecule that signals cells to do a thing. Depends on what the specific steroid is as to what the thing is. And the base molecule for all of our steroids is cholesterol. So our livers make cholesterol, and then that cholesterol gets converted into all these different steroids, which function as hormones. Again, hormones is a function because we do have peptide hormones, hormones that are made of protein. Okay, so it's a function. Steroid is the structure. And they're real easy to tell because of the fused rings. Okay, and then they have all these different functional groups. So this is a diagram showing you how cholesterol can be converted into all of these different hormones. And it's even more complicated than what you just know from like common knowledge, like how estrogen is for women and testosterone is for men. Well, there's all these other different hormones that work along with it. Like you have cholesterol, which becomes pregnenolone, which becomes progesterone, which becomes 17 alpha hydroxyprogesterone, which becomes I can't even say them properly, androstenedione, which becomes testosterone, which becomes dihydrotestosterone. Okay, so you see these are enzymatic pathways. So even though progesterone and estrogen, you think of, oh, those are for women. Well, no, all of these hormones are found in men too. And the male hormones are found in women. It's just the amount that they are found in and how they are used is different. And that's all for our lipids, short and sweet.